Hello, everyone. This is Mr. Ventura again. We're going to take you through a photosynthesis lab for this week. And what we're going to do is we're going to do a special kind of a chemical reaction that's going to tell you whether the light reactions or photosynthesis are actually happening or not. This is what you would actually be doing in the lab if we had the ability to actually go into the school building. Now here we have the light reactions of photosynthesis. You see them on the screen. This is a review from what we had in the lecture. So I hope you looked at the lecture um, uh, video before we do this. There is one on photosynthesis and respiration, and it describes photosynthesis in detail for you. But this is a review. Now here we have a light source. Of course, the light source is the sun. And the sun is going to shine down onto the earth. It takes about 8 minutes for the light to reach here. And when it does, it hits some kind of a plant leaf. And inside the plant leaf, there are chloroplasts. Chloroplasts are the structures, obviously, in the leaf that do photosynthesis for the plant. Inside the chloroplasts, there are structures called thylakoids. On the thylakoid membranes, there is a chemical called chlorophyll. Now, chlorophyll A is the type of chlorophyll that actually absorbs the light. And when it does, and it's helped by chlorophyll B and carotene, but chlorophyll A is the main pigment. But when it does, the light excites an electron, actually two of them, and the electrons fly up into a higher energy level in the molecule. They get excited by the energy in the light. But now that the chlorophyll lost its two electrons, they have to be replaced or else photosynthesis can't continue. So the way the plant replaces the electrons is by splitting water. Water will split, and when that happens, the electrons in the hydrogen portion of the water, two of them, will actually go back into the chlorophyll A and replace the ones that the chlorophyll A lost when the light excited them. When you split water, of course, you release the oxygen, and the oxygen comes off as a waste product. And that is what we breathe in the air today. Since we took the electrons off the hydrogens, we create two hydrogen ions. These are hydrogens without their electrons, and they are positively charged. So what's happening here is that the plant is really a solar-powered water splitter. The electrons from the chlorophyll that were excited get accepted by a molecule on the thylakoid membrane. Remember, the gain of electrons is reduction. So we can say that this molecule here, the electron acceptor, has been reduced. It gained the electrons. It gained the energy in the electrons. Once that happens, the electrons go through a chain of series of reactions from one to the other, to the next, and it's almost like a hot potato going from person to person. As the electrons go from molecule to molecule to molecule to molecule, they're gradually losing their energy, and some of it's coming off as heat. But some of that energy that the electrons are losing can be used to manufacture the all-important ATP molecule. ATP molecule is one of the products of the light reactions of photosynthesis. We're going to be using that in the dark reactions, or the Calvin cycle, to make sugar later on. Once the electrons get down to the last electron acceptor, it goes into another chlorophyll molecule. And this chlorophyll molecule does the same thing. The energy from the sun is going to excite the electrons again, and they're going to go jumping up to another electron acceptor. So there's two photosystems. Photosystem 2, which is the one that was discovered second, and photosystem 1, which is the one that actually happened second, but was discovered first. The electron acceptor that accepts from photosystem 1 here gives the electrons to a molecule called NADP. And this is what our lab is all about here. And this is the important portion. There's an enzyme called NADP reductase, which actually takes the electrons and gives them to the NADP. Once the NADP gets the electrons, it picks up the hydrogen ions from here, reunites the electron with them, and now you have a high energy hydrogen. That high energy hydrogen and the ATP is going to be used in the dark reactions to change carbon dioxide into sugar. But what we're going to do in our lab is we're not going to allow all the NADP here to accept the electrons. We're going to put a chemical in with the chloroplasts in the test tube. And that chemical is called DPIP. And you can see the DPIP is blue in color. When these electrons are supposed to go to the NADP, what happens is that the electrons instead are going to be picked up by our DPIP chemical that we put in there artificially in the test tube. When DPIP picks up the electrons, it changes to a clear color, and that'll look in the test tube a little greenish because the chloroplasts are still in there. So if we actually see the DPIP blue color disappearing, that means that you actually are watching photosynthesis happening, because those electrons are bouncing around from chlorophyll A to electron acceptors to a series of chemicals from one to the other down to chlorophyll A again, making ATP, going up to a electron acceptor the second time, and then instead of going to NADP where it's supposed to go, it goes to our DPIP chemical instead. They take the electrons. The DPIP is reduced, it accepts the electrons, and is changing to a clear color. And if we see DIP changing clear, that means that photosynthesis is actually happening. So how do we do the experiment? Well, if you look at this diagram on the bottom here, you see that we have some test tubes. We have four test tubes, there's 5 test tubes. You have one here, and these 3. All of these are the same test tube here. They're just different times. We're going to put some chloroplasts in this one, right here, this one, without any DPIP in it. So we'll see if that just stays green. That just shows everything is okay. In this one here, we're going to put DPIP, no chloroplasts. So that we expect that just to stay blue because there's no chloroplasts to give electrons to the DPIP, so they should stay blue. But in these 3 test tubes here, this one, and this one, we're going to put some chloroplasts and some DPIP. In this one here, we're going to put some live chloroplasts. We get them from spinach, and we're going to add some DPIP. We're going to put this test tube in bright light, and you can see after about 20 minutes, the blue color is disappearing. Okay, that's an indication that the DPIP here is accepting the electrons from the second electron acceptor here, and light reactions are actually happening. Now, you can actually measure how long it takes for that to turn clear, which will give you an indication of the rate of photosynthesis. But in our experiment here, we're just basically watching it happen. In this one, we boil the chloroplast first. Now, when you boil the chloroplast, and then add DPIP to them, and then shine a bright light at it, you don't expect too much to happen, because when you boil the chloroplast, you destroy most of the molecules and the enzymes in there, and so photosynthesis doesn't happen. And so we just, if photosynthesis is not going to happen, the DPIP will not be reduced. It won't be able to gain this electron, and therefore just stay blue. In the last one on the bottom here, you can see that what we did is we put it in the dark. And of course, without light, the photosynthesis doesn't happen either. And so you see the DPIP stays blue. So our experiment was a method to measure, or at least look at, the light reactions of photosynthesis actually happening by transferring electrons from chlorophyll all the way to DPIP instead of NADP. DPIP is the blue color we added to it. They steal the electrons from the second electron acceptor here, and they makes the DPIP turn clear. That's a way that you can actually show the photosynthesis actually happening. That's our lab. Okay, now there's going to be a short lab quiz about this. So, click on that when you feel comfortable, and take it for this week. Hope that helps you.